Hello everyone and welcome to the new session of the biology lecture series, and today we'll be discussing the NCERT Exemplar questions for the topic molecular basis of inheritance. So this topic is included in the class 12th NCERT biology book. Alright, and we have already solved some of the questions in the previous session. We have solved the multiple choice and the very short answer type questions, and today we'll be discussing the short answer type questions. So let's start with the first question. So here we have to define the transformation experiment done by Griffith. Okay, and you have to discuss how it helps in the identification of DNA as the genetic material. So you already have studied about the Griffith experiment. He was working on a bacteria or pathogen known as Streptococcus pneumoniae, and this causes pneumonia. Okay, so he was working on Streptococcus pneumonia. Okay, all right. And what disease does it cause? It causes pneumonia. So what he did, he infected, okay, with this bacteria in the mice, or he injected this bacteria in what? In mice. Okay, now first let's study what was Griffith experiment. So in the first experiment, what he did, he injected. Okay, first I will tell you about the strains of the pneumonia, Streptococcus pneumonia. So there are two strains of the Streptococcus pneumonia. First one is the S strain, also known as the smooth. Okay, it forms smooth colonies. Why it forms smooth colonies? Because it has a mucilaginous covering. So this S strain, this is the virulent strain. This is the virulent strain, all right, means it can cause disease. Okay, and if it causes diseases, okay, when injected in mice, mice will be dead. Okay, and there is another strain known as the R strain, rough strain. It forms rough colonies. It doesn't have a mucilaginous layer around it. So this strain is non-virulent. This is the non-virulent strain, means it doesn't cause any disease. All right. So if injected in mice, mice is alive. Okay, you can write here mice. Okay. Now, so what was Griffith's experiment? So what he did? In the first experiment, he injected the living S strain of bacteria. So this was living. He injected this in mice. And what he observed, as this is the virulent strain, the mice was dead. Now, in the second experiment, what he did, he injected the R strain of bacteria, okay, which was again living, in the mice. And in this case, mice was alive because this is the non-virulent strain. Then, in third experiment, what he did, he heat killed this S strain. Okay, so he injected the heat killed S strain into the mice. And in this case, what he observed, okay, mice was alive, was not dead. Okay. Now, in fourth experiment, what he did, he injected the mice with the heat killed S strain along with the R strain. okay, living R strain. He injected a combination of this, and what he observed? So R strain, you know, is non-virulent, and heat killed S strain also becomes non-virulent. So what happened here? Here, the mice was dead. Okay, now you can see why the mouse is dead here, because only living R strain is present here and the S strain has been heat killed. So why the mice is dead? All right. So based on this experiment, a transforming principle was discovered, and it was said that DNA is the transforming principle. So Griffith concluded that there is some transforming principle. There is some transforming principle, okay, that is injected or that is transferred from the S strain to the R strain, and the R strain was converted into the S strain. Okay, so when later on he isolated the bacteria from it, it was found to be living R strain bacteria was found. Okay, so this means that the R strain was transformed into the virulent S strain, and there was some transforming principle that was transferred from the S strain to the R strain. Okay, so this was Griffith's experiment. All right. So now in Griffith transformation experiment, okay, Griffith experiment can be defined as a change in the genetic constituent of an organism by picking up the DNA from the environment. Okay, Griffith experiment tells you about that the genetic constitution of an organism, okay, can be changed, or the organism can be transformed by picking up DNA from the environment or from the dead organism. So in this case, DNA from the R strain, which is known as the transforming principle. Earlier, it was not known that DNA is the genetic material. So he only said that there is some transforming principle. So this transforming principle was transferred from the S strain to the R strain, and then genetic constitution of the R strain changed and they become virulent S strain. Okay. Now, the transformation experiment played a role in identification of DNA as the genetic material. When heat was used to kill the virulent bacteria, means the S strain. I've already told you, when the heat killed S strain was injected in mice, then the mice was alive. They, okay, died, means the S strain bacteria died, not the mice. Okay, the S strain bacteria died, but their genetic material was still intact. All right, and that was transformed to the R strain. Now, this DNA, when picked up by the non-virulent bacteria, means the R strain, okay, made them capable of causing infection, means the R strain was transformed into S strain. Okay, this is clear. Now, Griffith concluded from his observation that R strain bacteria had undergone transformation by the S strain bacteria, and it was inferred that the R strain acquired a transforming principle from the heat-killed S strain bacteria, rendering them virulent. Now, this transforming principle was hypothesized to be the genetic material. Okay, so there was some transforming principle. Earlier, it was not known that it was DNA, but it was hypothesized that this is the genetic material. Okay, now question number 2. Who revealed the biochemical nature of the transforming principle? Okay, means biochemical nature means whether it is DNA. What is the biochemical nature? Whether it is RNA, whether it is proteins, or it is carbohydrates. What is the transforming principle? or what is the biochemical nature of the because if you talk about bacteria, it consists of nucleic acid. Okay, it can be DNA, RNA. It is also made up of proteins and carbohydrates, lipids also, which is present in the membrane. Okay, so let's discuss this question. So, Avery, McLeod, and McCarthy, so they were the scientists who revealed the biochemical nature of the transforming principle. Okay, so what they did, they were working on the extract of the heat-killed estrine. Okay, so what they were working on, they worked on the extract. They took the extract of estrine that were heat-killed. All right, this is clear. Now, what they did, first of all, in the first experiment, they made this extract, and from the extract, they removed the polysaccharide. So I'm writing cross for the removal. First, they removed the polysaccharides or the carbohydrates. Okay. Polysaccharides are carbohydrate. So you can use amylase, okay, to get rid of the carbohydrate. Amylase is an enzyme. Then in the second setup, they got rid of the lipids. And what they did, and they injected this extract in mice. This also in mice. In this case, what will happen? Mice will be dead or alive. In this case, the mice will be living. Okay, you observe that the mice was living. In this case also, mice is living. Then he got rid of the RNA. Here, to get rid of lipids, you can use lipase enzyme. To get rid of RNA, you can use RNase. And then he injected that extract in mice. Then again, the mice was living, it was alive. Then he got rid of what? Proteins. So you can use protease to get rid of proteins. Then he again injected that in mice. Again, the mice was living. Okay. So along with this, R-strain bacteria was also injected. Okay, along with this extract. And in the end, he got rid of the DNA using DNase. And then he injected that in mice, and he observed that the mice was, okay, it was living. So the experiment was done by Avery, McLeod, and McCarthy. So they revealed the biochemical nature of the transforming principle. So what they did, they were working on the extract of heat-killed estrine. Okay. Now what happened? They injected this extract along with the R-strain into the mice. So what in the first setup, what they did, they got rid of the polysaccharides, or you can say carbohydrates. Okay, so you can use amylase to get rid of carbohydrates. And then they injected that extract in mice along with the R-strain. And in this case, what was observed? The mice died. And what was obtained from this died mice? Living estrine bacteria were recovered. Again, in second setup, what they did, they got rid of proteins. Okay, so you can use protease enzyme to get rid of proteins. Then they injected that in mice. And what they observed? Again, the mice died. Again, estrine bacteria was recovered. Now in the third one, okay, they got rid of lipids. And then they did the same thing. They injected that in mice. Again, the mice was dead and estrine bacteria were recovered from the dead mice. Now in the next setup, they got rid of RNA using RNase. Again, they did the same thing, injected that in mice. Again, the mice was died, it was dead, and estrine living bacteria were recovered. In the last experiment, what they did? They got rid of the DNA. And then what they observed? The mice, okay, it was living and no living estrine bacteria was recovered. Okay, so now this experiment proved that DNA is the transforming principle. DNA is the genetic material. Okay, because when no DNA was present, okay, estrine bacteria were not recovered, which is virulent, and the mice was living. Okay, so DNA is the genetic material which is passed on to the next generation. Okay, now what did every McLeod and McCarthy did? They, okay, you can call their experiment was an extension of the Griffith experiment. So what they do? They did in vitro, means artificial studies. They created an in vitro system in order to determine the biochemical nature of the transforming principle. This already I have told you. Now, they reported that DNA from the heat-killed S-type bacteria caused the transformation of the non-virulent R-type bacteria into virulent S-type bacteria. They also discovered that proteases and RNase, as well as you can write lipases, amylases. Okay, you can add here protease, RNase. Protease, they degrade the lipids. RNase, they degrade the RNA. Then along with it, you can write lipases, okay, and amylases. So these enzymes, they did not affect the transformation, while DNAs, which degrade the DNA, inhibit the process of transformation. So they concluded that DNA is the heredity material. Now, question number 3. Discuss the significance of heavy isotope of nitrogen in Messelson and Stahl experiment. So, heavy isotope, you know, Messelson and Stahl, they grew the E. coli bacteria. So, they were working on the bacteria known as E. coli. Alright, and they used the heavy isotope of nitrogen, which was N15. Okay, so initially, they grew the E. coli bacteria in N15 containing media. Okay, for several generations. Okay, and later on, what they found that both the strands of the DNA has double strand, it is double stranded. So both the strand had nitrogen, or they had the heavy isotope of the nitrogen. Okay, this is clear. And then what they did? They transferred this bacteria on a N14 containing media. Okay, here you can write here, they transferred this bacteria, or they grew this bacteria into N14 containing media. Okay, and then what they observed? Okay, in the DNA, one strand was N15 and another strand was N14. Okay, so this shows the semi-conservative nature of the DNA. This shows semi-conservative nature of DNA replication. So Messelson and Stahl, they proved the semi-conservative nature of DNA replication. Here you can see one is parental and one is the newly synthesized. This is known as semi-conservative nature of DNA replication. So let's talk about this. Messelson and Stahl, they use heavy isotope, okay, which is N15. in the nutrient media to grow E. coli bacteria for several generations. So initially both the strand, okay, they consisted of heavy isotope of nitrogen, means N15 and N15. Now, they performed experiments on E. coli to prove that DNA replication is semi-conservative. So first what they did, they grew the bacteria in a medium containing, okay, N15, heavy isotope of nitrogen. So they used ammonium chloride, okay, in which the nitrogen was heavy. Heavy isotope of nitrogen was used, okay, in which heavy isotope N15 is the heavy isotope of the nitrogen, and they grew this bacteria for many generations. Then what they did, then they transferred the cells into a media with normal ammonium chloride, okay, in which N14 is the lighter isotope or the normal isotope. And then they took the samples at various definite time intervals as the cell multiplied. So they took the cells, okay, so generation 1 and then generation 2, okay. So you know the life cycle of E. coli bacteria is 20 minutes. So after 20 minutes, they isolated the bacteria and then they did the centrifugation in the cesium chloride, and they saw whether the DNA replication is semi-conservative or not. Now, the extracted DNA was centrifuged and measured for their densities. Okay. Now, the DNA extracted from the culture after one generation. Okay, one generation means after 20 minutes. Okay. Now, after one generation of the transfer from the N15 media to the N14 media, okay, that is after 20 minutes. E. coli divides, you know, after every 20 minutes. Then what was observed? Intermediate, or you can say hybrid density was observed. What does hybrid mean? That it both heavy and light nitrogen were present in the DNA, which proves that the semi-conservative nature of the DNA replication. Okay. So one of the strand after the first generation, one of the strand was or consisted of N15, another consisted of N14. Okay. So this shows the semi-conservative nature of DNA replication. Now let's talk about question number 4. Now here you have to define a cistron and then you have to give examples differentiating between monocistronic and polycistronic unit. Okay, now what is cistron? Cistron is a RNA or DNA sequence that code for a particular polypeptide. So what is a cistron? In simple words, you can define it is a DNA or a RNA sequence, all right, that codes for a specific polypeptide. This is the definition of cistron. Now, if we talk about eukaryotes, in eukaryote we have monocistronic genes or monocistronic mRNA. Okay, if you talk about polycistronic unit, these are present in prokaryotes. Okay, so prokaryote consists of polycistronic genes or polycistronic mRNA. So what is monocistronic genes? Where a single cistron is present. Okay, if we talk about monocistronic genes, it has only one cistron. Okay, or you can say it consists of only one structural gene. It consists of only one structural gene, means it codes for only one protein or polypeptide. Then what about polycistronic? Okay, it consists of many cistron. All right, or you can say it consists of more than one structural genes. Okay. If it consists of more than one structural gene, then it consists of more than one polypeptide or protein. All right, this is clear. All right, let's discuss about the answer. Now, a cistron is a stretch of base sequences that codes for one polypeptide chain. Okay, and it also includes the adjacent control regions, which are the promoter and the operator. Now, it may also code for the transfer RNA, ribosomal RNA molecule, or it may perform other specific functions, including regulating functions of other cistrons. Okay, this is known as cistron. Now, this term has replaced the definition of a gene. Now, monocistronic transcription unit will have all the regulatory and the coding sequences for a single polypeptide. Okay, I've told you it consists of a one cistron, means it codes for only single polypeptide. Okay. Now, whereas the polycistronic units such as the lac-operon may have coding sequences for more than one polypeptide. So, example of a polycistronic unit is lac-operon. You must have studied about lac-operon. It consists of the regulatory sequences. Okay, it consists of a promoter. Okay, then it consists of an operator. Then lac-operon also consists of a regulatory gene, which synthesize the repressor molecule. And along with this promoter, operator, and regulatory gene, so these are the regulatory sequences. Okay, we have the structural genes as well. Structural genes. Okay, so in lac-operon, we have Z gene, X gene, okay, Z gene, Y gene, and A gene. Okay, one codes for beta galactosidase, and then other code for transaminase, and the other codes for, okay, permease. All right, so this was about monocistronic and polycistronic unit. Now, in eukaryotic cells, almost all the messenger RNA are monocistronic. All right, so in prokaryotes, monocistronic units are present, whether in prokaryote, such as lac-operon, coding sequences would be an example of a polycistronic DNA region. Now, coming to question number 5. Okay, now here you have to give any 6 features of the human genome. So, human genome has been sequenced. Okay, it was sequenced during the Human Genome Project. That was a collaborative effect. A lot of countries was involved. All right. So here you have to list the features of the human genome. Now, first one, what are the salient features of the human genome? First one, the human genome contains 3,164 million nucleotide bases. Okay. So it is very large. Then the average gene consists of around 3,000 bases. So if you talk about an average gene, it consists of about 3,000 bases. And the largest known human gene, okay, is known as dystrophin, which is at 2.4 million bases, or we can say which is located at 2.4 million bases. And in total, we have 3,164.7 million nucleotide bases. And the total number of genes is estimated to be 30,000. Okay. And 99% of the nucleotide bases are exactly the same in all the people. All right. This is a very interesting feature. 99% of nucleotide bases are exactly same in all the people. Only 0.1% differences can be seen. Okay. Now, the functions of a lot of the genome are unknown. Okay. for over 50% of the discovered gene. Okay, now genes has been discovered, okay, but the functions are not known. Now the functions are unknown for over 50% of the discovered genes. We don't know the functions of these genes. Okay, what do they do? Now less than 2% of the genome codes for protein. So this is also a very interesting factor. Rest is the waste DNA or the junk DNA. Okay, and only 2% of the genome codes for protein, or you can say enzymes. Means only 2% of the genome, okay, is useful. Rest is also useful, but, or you can say rest of the genome, that is known as the junk DNA. Now, the human genome contains, okay, large repeated sequences. This is also a unique feature. The human genome, okay, it consists of large repeated sequences. Sequences are being repeated, okay, and we use this repetition for a lot of things. Okay, we will talk about this later. Now, these repeated sequences is thought to have no direct coding functions. Okay, so these repeated sequences, okay, they don't have any coding function, means they don't code for any proteins. Okay, but they have other functions. What functions? But they throw light on what? Structure of the chromosomes, dynamics, and evolution. All right, this is clear. And the chromosome 1, we talk about the chromosome 1 has most of the genes, okay, or the highest number of genes, which is 2968, and the Y chromosome, which is the male chromosome. So this is the male chromosome and it has the fewest number of genes, only 231. Okay, so this is also a very interesting fact. And in the end, scientists have identified about 1.4 million locations where single base DNA sequences differences occur. Okay, and these are known as single nucleotide polymorphism or single nucleotide, okay, base pair differences, and these occur in human beings. Okay, so see, okay, 1.4 million locations. At 1.4 million location, there is a single base pair DNA sequence change. Single base pair. Suppose, just for an example, we have GAG. Okay, instead of GAG, it is GTG. So there is a difference in only one base. This is known as single nucleotide polymorphism, or you can say single base DNA sequence differences. So this is clear. So these were the unique features of the human genome. It is very large. You can see the average number of genes. Okay, it's around 3,000. Total number of genes. Okay, they are 30,000, but we don't know the functions of over 50% of the genes. Only 2% of the genome is coding. That is codes for proteins and all. And rest is known as the junk DNA. Another feature is that the human genome, they consist of repeating units. And these repeating units, okay, are non-coding. But they have some other functions, like they determine the structure of proteins and all. Okay, and there are about 1.4 million locations where single nucleotide polymorphism exists. Okay, so all these features are useful in determining the parental, okay, disputes and all, everything. They are also involved in inheritance of the, or determining the inheritance of the genetic diseases and all. Okay, so this is clear. Now let's move on to the next question. So let's start with the sixth question. So the question is, during DNA replication, why it is that the entire DNA molecule does not open in one go? And you have to explain what is replication fork and what are the two functions of the DNTPs. Okay. Now, first of all, you must be knowing what is replication fork and how it is formed. Here, the enzymes involved are, first one is helicase. So helicase is the enzyme, okay, that unwinds the DNA. Okay, it unwinds the DNA, or you can say it unwinds the DNA strands. Okay, this is the role of the DNA helicase. Then what happens? single strands are created, and these single strands are stabilized by SSB proteins. Okay, so SSB protein comes, they bind to the single stranded DNA. Okay, they bind to the single stranded DNA, and then they stabilize the DNA, and the DNA they remain in the single stranded state. What is the full form of SSB? SSB is single stranded, okay, binding proteins. They are the protein molecules. Okay. Now what happens? As the helicase unwinds the DNA, then what happens? There is some tension created. Okay. This unwinding leads to a tension in the DNA molecule. And this tension is relieved by another enzyme known as topoisomerase. Okay. So a tension is created as the DNA unwinds, and this tension is relaxed, you can say. This tension is relaxed by an enzyme known as topoisomerase. Okay. Now, how this topoisomerase works? It creates nicks in the single-stranded DNA. Okay. And it relaxes the tension which is created by the uncoiling of the DNA with the help of helicase enzyme. Alright, so now this is clear. So here how replication fork looks like. Okay, suppose this is a replication fork. This is one strand of DNA. Okay, now this is another strand of DNA. Okay, so here this is the leading strand from 3' to 5'. So this will be the lagging strand, which will be from 5' to 3'. Okay, this is known as the leading strand, which has the polarity from 3' to 5'. This is the lagging strand. Okay, here continuous synthesis of DNA takes place. Okay, from 5' to 3'. Here discontinuous synthesis of DNA takes place. Okay, which will be in which direction? Here it will be 3'. So here from 5' to 3'. Okay. Now this is known as the replication fork. Okay, this is known as the replication fork, which is formed within the replication bubble. Okay, this is clear. Now here, replication fork is moving in this direction. This is the direction of movement of the replication fork. Okay. Now here, unwinding is taking place. SSB proteins come here, and then topoisomerase come here. Okay. So here is the direction in which replication fork is forming. Okay, so this is a very rough diagram of the replication fork that is formed within the replication bubble. Alright, this is clear. Now let's have a look at the answer. Now, during replication, the entire DNA molecule does not open simultaneously to maintain the overall stability, as this process requires significant energy expenditure. Now, this unwinding creates tension in the DNA molecule. This already I have told you. Okay, this unwinding creates tension in the molecule. As the uncoiling part, they start forming supercoils. Okay, so the uncoiled parts, okay, which are formed due to the unwinding, they start forming supercoils. And why they form supercoils? Due to the interaction of the exposed nucleotides. Now the nucleotides are exposed, so they can bind with each other and they form supercoils. Now, helicase enzyme act on the double strand, where they act on the ORI site, means origin of replication. Okay, so this you have to remember. Helicase enzyme it acts on the origin of replication, which is AT rich. Okay, why it is AT rich? Because A and T, they pair up using 2 hydrogen bonds, whereas GC, they pair up with each other with the help of 3 hydrogen bonds. So ori, they are usually AT rich. Okay, so they are easily to break these bonds. All right. Now, and a small stretch is unzipped. Okay, so slowly the DNA molecule is getting unwinded. Okay, so this takes place slowly, not the entire DNA molecule is getting unwind. Okay, immediately it is held and stabilized by single strand binding proteins. Okay, so when the single strand is created, then they are bound with the single strand binding proteins. And then what happens? Type 1 topoisomerase, they alleviate, means reduce the tension generated during the DNA binding and unwinding. So here we are concerned with unwinding. Okay, so the tension created due to unwinding is relaxed by the topoisomerase. Okay, that's why the entire DNA molecule doesn't open at one time, but it opens in small stretches. Okay. Now, replication fork. Replication fork gives an appearance of a Y-shaped structure. Okay, so this Y-shaped structure is known as the replication fork. I've already drawn the diagram of the replication fork. Now, what are the two functions that the monomeric units of DNT will play? Okay, first, they pair up with exposed nucleotides of the template strand and they make phosphodiester linkages and release a pyrophosphate. Okay, this is clear. And then what happens? Hydrolysis of this pyrophosphate by enzyme pyrophosphatase. Okay, so there is an enzyme pyrophosphatase. Okay, that carry on the hydrolysis of the pyrophosphate, and in this process energy is released, and that energy facilitate making hydrogen bonds between the free nucleotide and bases of the template DNA. So these are the functions of DNTPs. Now, next question is, retroviruses do not follow the central dogma. Now, first of all, what is central dogma? Central dogma is, okay, from DNA, RNA is formed, and from RNA, proteins are formed. Okay, so this process is known as transcription. Okay, and this process is known as translation. All right, now in retrovirus means viruses which contain RNA as the genetic material, such as HIV, such as tobacco mosaic virus, QB bacteriophage, influenza virus. These are all RNA virus. Here, a complementary DNA is made from the RNA. So here reverse is happening. From RNA, DNA is being synthesized, and this process is known as reverse transcription. This process is known as reverse transcription, and the enzyme involved is reverse transcriptase. Okay, so here from RNA, a complementary DNA is formed. Okay, so retroviruses, they do not follow central dogma, which is DNA to RNA to proteins, because the genetic material is not DNA. They have RNA as the genetic material. Instead, they have RNA that is converted to DNA. And which DNA? cDNA, complementary DNA, by the enzyme reverse transcriptase. Now let's move on to the next question. In an experiment, DNA is treated with a compound which tends to, so this DNA, okay, it is placed, okay, or it gets fixed where? Among the stacks of the nitrogenous base pairs. Okay, so what happens? So as a result of this, the distance between the two consecutive base pair increases from 0.3 to 0.4 nanometer. Now here you have to calculate the length of the DNA double helix, which has this much amount of base pairs in the presence of this saturation of this compound. Okay, Now you already know DNA. If we talk about the DNA is a double helical structure. Okay. These are the bases. Bases are here like this. Okay. A pairs with T, G pairs with C. Okay. Now this distance, this is 0.43 nanometer. Okay. This is the distance between the two nitrogenous base. Okay. Now this distance has increased to, I think, 0.44 nanometer. Okay. Because here some compound has come which has stacked itself or which has placed itself within the nitrogenous bases. Okay. Now you have to answer. Now let's have a look at the answer. So before solving this question, we need to know a formula. Okay. The formula is, okay, number of, if we have to calculate the length of the DNA, number of the base pairs present, okay, into the distance between two consecutive base pairs. Okay, this is clear. This is how we calculate the length of the DNA. All right, this is clear. Now, let's have a look at the answer. So, the length of the DNA double helix, you can calculate now. We have this much amount of base pairs. And now, from 0.3, the distance between the consecutive base pairs is 0.44 nanometer. So, it was 0.44 nanometer, which means 44 into 10 raised to the minus 9 meter. Okay, this is what is written here. So you can calculate and you can write the answer here, which will be around 0.8. Okay, 0.88. Now question number 9. What would happen if histones were to be mutated and made rich in the acidic amino acid? You know, histones are basically, they are positively charged. What are the features of histones? They are positively charged. Okay, and they are basic in nature. They are basic in nature. All right. Now, if they are mutated, it is saying, and they are made rich in acidic amino acids such as aspartic acid and glutamic acid in place of the basic amino acid, which are lysine and arginine, what would happen? Okay. Now, because histones are usually made up of basic amino acids, they are positively charged. And this helps the DNA to, okay, wind around them. So, if suppose these are, so you know, nucleosome, it is made up of 8 histones. It is made up of 8, or you can say it is made up of histone octamer. So, this is H2A, H2B, this is H3 and H4. Okay. Now, there are 8 of these and the DNA is coiled around it. Okay, DNA is coiled around this. So, the DNA, you know, is negatively charged because of the presence of the phosphate. Whereas histones, if we talk about histone, these are positively charged. Okay, so here you can see, if histones were mutated and made rich in acidic amino acid, they will not be able to serve the purpose of keeping the DNA coiled around them. Okay, so here negative charge is stabilized by the positive charge of the histones. So this is because DNA is negative charge because of the presence of phosphate, and histones are positive charge because of the presence of basic amino acids. Okay, so they are attracted to each other if the histones become negatively charged. Okay, instead of binding, they would rather repel the DNA. Okay. Now, the packaging of DNA in the eukaryotes would not happen. Consequently, chromatin fiber will not form. All right. So, it will disturb the nucleosome formation. And what is nucleosome? I've already shown you. It is made up of the histone octamer and the DNA is coiled around it. And how much DNA? 146 base pair of DNA is coiled around it. Okay, this is clear. Now, nucleosome, it is the structural and functional unit of chromatin. It is a structural and functional unit of chromatin. Okay, so that's why chromatin fiber will not form if nucleosomes are not formed. Now, coming to question number 12, here we have to recall the experiments done by the Griffith, okay, Avery, McLeod and McCarthy, where DNA was speculated to be the genetic material. Okay, so Griffith, he discovered the transforming principle, and Avery, McLeod and McCarthy, they further worked on the experiment, or they further continued the Griffith experiment, and they showed that the DNA was the speculated genetic material. Now, if RNA instead of the DNA was a genetic material, okay, would the heat-killed strain of pneumococcus have transformed the R strain into the virulent strain? So R strain, you remember, was the non-virulent strain. Okay, it doesn't cause any disease, it doesn't cause pneumonia. So what was happening in the last experiment when the heat-killed strain was added along with the R strain, and both of these were injected into the mice died, and the R strain were transformed into the S strain. Okay, but here in the question is saying if RNA is the genetic material, then what would happen? So, answer is RNA, you know, is less stable, it is more labile, and it is prone to degradation. Why it is unstable? Because of the presence of the 2' hydroxyl group, which is present in the ribose sugar. Okay, therefore, the heat-killed S strain may not have retained its ability to transform the R strain, okay, into the S strain, virulent strain, if RNA was its genetic material. So RNA would have been degraded because RNA is very unstable due to the presence of the 2' hydroxyl group. Now, coming to question number 11. Now, you are repeating the Hershey and Chase experiment. Okay, so Hershey and Chase experiment, they used, okay, sulfur and radioactive phosphorus, and you are provided with two isotopes, phosphate and the heavy isotope of nitrogen. So you are using heavy isotope of nitrogen instead of the sulfur. Okay, now how do you expect your results to differ? All right, now use of the heavy isotope nitrogen will be inappropriate because method of detection of phosphate and nitrogen are different. Okay, so this heavy nitrogen, this is non-radioactive, but phosphate, okay, this is radioactive. So their method of detection is first of all different. And even if the heavy isotope of nitrogen was radioactive, then also what happened? You know, DNA. DNA is made up of what? It is made up of nitrogenous bases. Okay, and it is made up of what? It is made up of the pentose sugar. And what else? It consists of a phosphate group. Okay, it also consists of a phosphate group. This is clear. Now, proteins also. Proteins also are made up of nitrogen. So they have the amino group. Okay. They have the carboxylic group also. Here in the center, carbon is attached. One will be H. This is the alkyl group, which is the variable group. So here you can see nitrogen can be incorporated in proteins. Nitrogen can be incorporated in DNA also. So even if the nitrogen was radioactive, then its presence would have been detected both inside the cell, means in the DNA, okay, as well as in the supernatant, which consists of the proteins. Okay, so N15 would also get incorporated in the amino group of the proteins. Hence, the use of N15 would not give any conclusive result, because N15 will be incorporated in both proteins and the DNA. So it will be present inside the cells also, okay, in the DNA, and it will be present in the supernatant also, where proteins are present. Okay. Now, question number 12. There is only one possible sequence of amino acid when reduced from a given nucleotides. Okay, so this is clear. But what happens? Multiple nucleotide sequences can be reduced from a single amino acid sequence. Now you have to explain this. Okay, now let's talk about the answer. Now, some amino acids are coded by more than one codon. This is known as degeneracy of codons, where more than one codon codes for a single amino acid. Okay, this is known as degeneracy of codons. So, you know, we have 64 codons. So, codons, they are made up of 3 bases. Okay, so and in total we have 4 nitrogenous bases, and codon consists of 3 bases, okay, such as AUG. So in total we have 64 codons, but they code for only 20 amino acid. So they code for only 20 amino acids. Okay, so you can say that more than one codon code for a single amino acid. So hence, on reducing a nucleotide sequence from an amino acid sequence, multiple nucleotide sequence will be obtained. For example, isoleucine. Isoleucine has 3 codons: AUU, AUC, and AUA. Okay, so this isoleucine amino acid is coded by these 3 codons. Hence, a dipeptide, methionine. Suppose we are taking example of a dipeptide which consists of methionine and isoleucine. They can have the following nucleotide sequence. So isoleucine, we have already seen, is coded by 3 codons. So what can be the nucleotide sequence of this dipeptide? Methionine, you know, is coded by AUG. And isoleucine, you have seen, can be coded by AUU. It can be coded by AUC and can be coded by AUA. Alright. So if we reduce amino acid sequence from the above nucleotide sequence, all the 3 will code for methionine and isoleucine. Now coming to next question. A single base mutation in a gene may not always result in loss or gain of the function. Now you have to explain, do you think the statement is correct? And you have to define your answer. Okay. Now, first of all, this statement is correct. Now, what is the explanation of this? Now, because of the degeneracy of the codons, we have already studied that. What is degeneracy? It means more than one codon codes for a single amino acids. So, because of degeneracy of codons, mutation at the third base of the codon. Okay, so codon, it consists of 3 bases. So, this last base, this is known as the wobble base. So this base, okay, show a lot of variation. Okay, so the third base of the codon, known as the wobble base, usually does not result any changes in its phenotype. So if there is a change in the third base, okay, so it, okay, usually will not cause any change in the phenotype. Means it will code for the same protein, you can say, or same polypeptide, same amino acid. Okay, and there will be no change in the phenotype. So these kinds of mutations are known as silent mutation. Also, there is a change at the third base, but there is no change in the phenotype, because that codon still code for the same amino acid. Now, on the other hand, what happens if the codon is changed in a way that now it specifies another amino acid? Okay, so we have seen the example of sickle cell anemia. In that, the beta globulin chain of the amino acid, there is a change in the 6th position of the beta globulin chain. Okay. So here what happens? Glutamic acid. The glutamic acid changes into, or it is substituted by, valine. So glutamic acid is known as, or GAG codes for glutamic acid, but there is a single nucleotide polymorphism. There is a change at the single base pair, and then it codes for GTG. And you can say GTG codes for valine. So here is a change in the amino acid, and this causes, okay, a change in the shape of the erythrocytes. So this is what happens in the sickle cell anemia. So if the codon change in a way that it specifies for another amino acid, it may alter the protein function, as it happens in the case of beta globulin hemoglobin protein, where a substitution of valine instead of glutamic acid causes change in its structure and the function, resulting in sickle cell trait. Now moving on to the next question. A low level of expression of lag operon occurs all the time. Okay, can you explain the logic behind this phenomena? Why the low level of expansion of lag operon occurs all the time? So lag operon, okay, it helps in the metabolism of the lactose. It is involved in the metabolism of lactose. Okay, so in the presence of the lactose, when lactose is present, okay, so lactose acts as a inducer. And then what happens? In the presence of the lactose, there is the transcription of the structural genes. So I'm writing tick mark, means the lag operon is active. Then when the lactose is absent, then you already know the repressor molecule, it can bind to the operator, forming repressor operator complex, which inhibits the transcription of the structural genes, and then translation is also inhibited. So lactose, when is absent, okay, then what happens? Lag operon is switched off, or it is not active. Here the lag operon is active. So this is clear. And lag operon, it consists of 3 structural genes, okay, that code for beta galactosidase, permease, and transacetylase. Okay, now in the complete absence of expression of the lag operon, permease, which is the enzyme produced, which is one of the enzymes produced by the lag operon, this will not be synthesized, and this permease is essential for transport of lactose from medium into the cells. Okay, and if lactose cannot be transported into the cells, then what will happen? Then it cannot act as inducer. Okay, so lactose is an inducer. Hence, okay, it cannot relieve the lag operon from its repressed state. So when lactose is present, then what will happen? Lactose will bind to the repressor, which I am writing as R. So when the lactose binds with the repressor, then this complex now cannot bind to the operator, and the lag operon is switched on. Okay, so this is clear. Moving on to the next question. How has the sequencing of human genome opened new windows for treatment of various genetic disorders? Now you have to discuss this question. So here I've given you a thinking process here, just a brief idea about Human Genome Project. Now in 1990, US Department of Energy and National Institute of Health They embarked and they coordinated on the project of sequencing of human genome. And this project was known as Human Genome Project. Okay, so this was a collaborative effect within many countries. Now, the sequencing of human genome helped in enhancing the basic understanding of genetics and immunity to various disorders. Okay, now we know in Human Genome Project, we know that how many genes are present in humans. Okay, and we know the functions of not all the genes, but we know the functions of the genes now, and we know at which position a gene is located. We know that which chromosome has how much number of genes. Okay, now various genes that cause genetic disorders were identified with the help of this project. Okay, but still over 50% of genes, okay, or you can say the functions of over 50% of gene is still unknown. All right. Now, it was found that more than 1200 genes are responsible for human cardiovascular diseases, endocrine diseases such as diabetes, neurological disorders like Alzheimer's disease, cancers, and many more. Now, these diseases can be treated easily by knowing the particular gene responsible for the particular disease. Okay, if we know which gene is responsible for this disease, so we can manipulate it, we can correct it, and then we can treat the disease. Now let's move on to the next question. The total number of genes in humans is far less than 25,000 than the previous estimate. So previously it was estimated that human genes or human genome consists of 1,40,000 genes. But in the end, after the Human Genome Project, it was observed that only around 25,000 genes are present in human genome. What is the reason for this? Now, the total number of genes is estimated to be 25,000, which is much lower than the previous estimate of 1,40,000. Okay, so this estimate was based on the extrapolation of the gene-rich areas. So they were only considering the gene-rich areas. Okay, so they extrapolated it. Okay, they were only focusing on gene-rich areas. Okay, but actually in the human genome, we have a composite of gene-rich as well as a gene-poor areas. Okay, so they ignored this gene-poor areas. All right. Now, some other facts about human genome. Almost 99.9% nucleotide base, they are exactly the same in all the people. And functions of over 50% discovered genes are not yet known. Okay, this already I've told you. Now, scientists have identified about 1.4 million locations where single base pair DNA differences or single nucleotide polymorphism occur in humans. Okay, so all this information is very essential. It can help treat diseases. It can help to know the inheritance pattern of the genetic related diseases. Okay, and many more things. Now, this information promises to revolutionize the process of finding chromosomal locations for disease-associated sequence and tracing the human history. Okay. Now, let's move on to the next question. Question number 17. Now, sequencing of total genome is getting less expensive day by day. Soon, it may be affordable for a common man to get his genome sequenced. What is your opinion? Could be the advantages and disadvantages of this development. So, what are the advantages and disadvantages? Now, human genome helps to find out the complete genome sequence of the human, and it may have following advantages and disadvantages. So, first, let's have a look at some of the advantages. Now, the knowledge of the effects of variations of DNA among the individual can revolutionize the ways to diagnose, treat, and prevent many diseases that affect the humans. Okay. It also provides clues to the understanding of human biology, and it helps to find out the human evolution. Identification through DNA forensic is also possible due to the sequencing of the human genome. Okay. Now, what are the disadvantages? Now, people might discover untreatable genetic diseases. Okay. This is the first disadvantage. Second is people may abuse the knowledge obtained from the Human Genome Project. Okay. They can create some superhuman or something like that. Okay. Or they can use it for some other purpose. Now, the problem can occur for the ownership of the genetic test, okay, results and the patenting of human genes. This is known as it's kind of a biopiracy only. So they are, okay, they can patent the genetic test results, okay, and they can also patent the human genes and the DNA, which is illegal because this is natural, okay. So this condition can arise. Now, people believe that they are special and unique in their own ways and may wish to remain like that. So, okay, so now we know the sequence of the human genome. We know the genes and all. We know which genes consist, which chromosome consists, how many number of genes, and also we can now manipulate the human genome and we can create recombinants. We can manipulate and then we can create recombinants. Okay, so here we are playing with the nature. Okay, but people, what they believe that they should remain natural only. Okay, so this is like acting like God. We can manipulate and we can make a individual that has all the genes that we wish. Okay, so these are some of the disadvantages. Now, question number 18. Would it be appropriate to use DNA probes such as VNTR in DNA fingerprinting? Now, what are VNTRs? VNTR are variable number of tandem repeats. Okay, variable number of tandem repeats, and they are short nucleotide sequences. Okay, around 20 to 100 base pairs. Okay, so these are very short nucleotide sequences. Okay, that vary in copy number. Okay, so the copy number is different in different individuals. And because of this, okay, a unique pattern is observed. For every individual, there is a unique pattern is observed. All right, this is clear. And where are VNTRs? What is the location of where they are present? They can be present in exons. They can be present in introns, as well as they can be present in intergenic regions, means within in between the genes. Okay, now this is clear. I will also like to tell you about what are the functions of this VNTR. So VNTR, they can be used as RFLP genetic markers. So they can be used as the genetic markers. And so this is the first role. Second role, they can also be used in the mapping of the human genome. Okay, mapping of the genome, you can say mapping of any genome. Okay, and because VNTRs, they form the unique pattern, they can also be used to determine the individuality of the organisms. Okay, so this is clear. Now let's talk about the answer. Now, bacteriophages does not have any repetitive sequences. So VNTRs, they are repetitive sequences which vary in the copy number. Okay, so bacteriophage do not have any repetitive sequences and their genome is very small and have only the coding sequences. Okay, so these VNTRs, they don't have any function. You can write they don't have any coding functions. Okay. So the bacteriophage DNA is very small and it consists of only the coding sequences. Okay. DNA fingerprinting is not done for bacteriophages. So that's why we cannot apply the technique of VNTR for bacteriophages. We cannot apply the technique of VNTR for bacteriophages and DNA fingerprinting using VNTR. So what is the basis of DNA fingerprinting? It is based on VNTR. Okay, because it forms the unique pattern of any individual. Because every individual, they have different copy number of this VNTR, and this VNTR are usually, they don't have any function. They are present in the non-coding regions such as introns. Okay. In bacteriophage, they have only coding sequences. The question number 19. During in vitro synthesis of DNA, a researcher used 2' to 3' dideoxycytidine triphosphate as raw nucleotide in place of 2-deoxycytidine. Now, what will be the consequences? So, answer is, now the further polymerization would not occur as the 3' OH on the sugar is not there to add new nucleotide for the formation of the ester bond. So, the new nucleotides are added at the 3' end. So, the new nucleotide, it is added at the 3' end. So, the polymerization would not occur as the 3' OH on the sugar is not there to add the new nucleotide for forming the ester bonds. Now, question number 20. The background information did Watson and Crick have made available for developing a model of DNA, and what was their contribution? Okay. Now, Watson and Crick had the following information. Okay. What information they had? So Watson and Crick, they gave the double helical structure of the DNA, and they had some background information which helped them to develop the model of DNA. What was that background information? First of all, Chargaff. Okay. So they had the information of the Chargaff. So what was Chargaff law? That the amount of adenine is equal to amount of thymine and amount of, okay, here it should be triple bond. Amount of cytosine is equal to amount of guanine, and the ratio of adenine by thymine is equal to ratio of guanine by cytosine, and this ratio is always 1. Okay. So Watson Crick had this background information. Okay. Now, second, they used Wilkins and Rosalind Franklin's work, okay, on DNA crystals X-ray diffraction studies about the DNA physical structure. So Wilkins and Franklin, okay, they both provided, okay, some information. What was that information? Okay, they provide the X-ray diffraction studies. They provide this X-ray diffraction studies, okay, about the DNA physical structure. What is the physical structure of the DNA? So Watson and Crick also had this information. Okay, so based on this, they developed the double helical model of the DNA. Now, what is Watson and Crick proposed? First of all, what they proposed? They propose a pattern of the complementary base pairing. Means A pairs with T and G will pair with C, and G pairs with C with the help of 3 hydrogen bonds, and A pairs with T with the help of 2 hydrogen bonds. Semi-conservative replication. Actually, Messelson and Stahl proposed the semi-conservative replication, but Watson and Crick has also some role in proposing the semi-conservative replication. Okay, but it was Messelson and Stahl, okay, who gave the semi-conservative nature of the DNA replication. And Watson also proposed that mutation occurs through tautomerism. Now let's talk about question number 21. What are the functions of first methylated guanine cap and poly A tail in a mature RNA? So there is a process. First of all, from DNA, what is formed? hnRNA is formed. Okay, so hnRNA, it consists of both introns, it consists of exons also. All right. And then there is the processing, and then the mature RNA is formed. And this mature RNA, okay, it has a 5' and the 3' end. At the 5' end, guanine cap is added. At the 3' end, poly A tail is present. Okay. Now we have to tell the functions of this. So the methylated guanine cap helps in binding of the mRNA to the small ribosomal subunit. Okay, so this you have to remember. The small ribosomal subunit, okay, it adds at the guanine cap, 5' guanine cap during the initiation of translation. Okay, so this is the function of the guanine cap. And what is the function of poly A tail? So poly A tail provide longevity to the mRNA's life. Okay, if poly A tail is not present, then mRNA will be degraded. Okay, and also poly A tail acts as a signal. Okay, so initially the RNA or the transcription takes place in the DNA in nucleus in eukaryotes, and then the RNA is processed and then it's transferred to the cytoplasm where translation takes place. So poly A tail, okay, acts as a signal that okay, now the RNA is ready to be transferred. Okay, it has been processed. So poly A tail provides longevity to the mRNA's life. The tail length and longevity of mRNA are positively correlated. Okay, so poly A tail, one function is it provides the longevity, and second is after the formation of poly A tails, now cells get the signal that now it is mature and it can be transported into the cytoplasm from the nucleus. The next question is, do you think that the alternate splicing of exons, okay, so intron splicing is different, we are talking about alternate splicing here, may enable a structural gene to code for several isoproteins from one and the same gene? If yes, you have to tell how. Now, the functional mRNA of the structural genes need not always include all of its exons, okay, because of alternate splicing. So this alternate splicing of exon is sex specific, it is tissue specific, and even the developmental stage specific. Okay, so according to the developmental stage, according to sex, and according to the tissue, okay, there is splicing of the exons. So exons, are the coding sequences. They are the coding sequences, means they code for a particular polypeptide or the protein. But due to alternate splicing, different proteins can form. Okay, so initially if there are 5 exons, due to intron splicing, 1 exon is removed, only 3 is left. Okay, now by such alternate splicing of exons, a single gene may encode for several isoproteins or proteins of the similar class. Okay, so if exon 1 is present, then exon 2 is present, exon 3 is present, exon 4 is present. So after alternate splicing, you can see, suppose exon 1 is left, then exon 3 is left, then exon 4 is left, and this will code for a particular protein. Okay, and that's how we get different isoproteins or proteins of similar class. Now, in the absence of such kind of splicing, there should have been new genes for every protein. Okay, so if alternate splicing is not present, then for every protein a new gene is required. Okay, such an extravagancy has been avoided in natural phenomena by way of alternate splicing. So this is an advantage of alternate splicing. Otherwise, a new gene will be required for every protein. Now, question number 23. Comment on the utility of variability in number of tandem repeats during DNA fingerprinting. So, I've already told you, DNA fingerprinting is based on VNTR, variable number of tandem repeats. Okay. Now the randomness in repeats provides many copies of the sequence for fingerprinting and variability in the nitrogen-based sequences present in them. Now, being individual specific, these proves to be useful in the process of DNA fingerprinting. So, I've already told you VNTR, okay, they are short DNA sequences and they vary in the copy number. They vary in copy number, and because of this, they form very specific patterns. They form very specific patterns, and this technique or VNTR forms the basis of the DNA fingerprinting. And I've already told you about the function or the utilization of the VNTRs and where VNTRs are present. Okay, so that's why VNTR or the randomness or the tandem repeats is an advantage in DNA fingerprinting. So here again, randomness in repeats, okay, provides many copies of the sequence for fingerprinting and variability in the nitrogenous base sequences present in them. All right, so this is clear. So this was the end of the session. We will start with long answer type questions in the next session. Thank you very much for listening.